Hello friends, today we will gain knowledge about concept of haemopoesis, hemostasis which includes all types of clotting mechanisms and basic concepts of transfusion. And our main objectives will be concept of haemopoesis and its process. Second, to study about hemostasis and all types of clotting mechanisms. And third, to know about the basic concepts of transfusion. First, let me introduce the term haemopoesis. It's a process which includes origin, development and maturation of all the blood cells. The stem cells are the primitive cells in the bone marrow which give rise to all the blood cells. Stem cell is defined as a cell which is capable of both self-renewal and differentiation. Pluripotent hemopoietic stem cells are derived from the stem cells. These are defined as a cell that can give rise to cells of all groups of hemopoietic cells like myeloid and lymphoid cells. In the early stages, the PHSC are not designed to form a particular type of blood cell, and also it's not possible to determine the blood cell to be developed from these cells. So, at this stage, the PHSC are called uncommitted pluripotent hemopoietic stem cells. When the cells are designed to form a particular type of blood cell, the uncommitted PHSC are called committed pluripotent hemopoietic stem cells, which is defined as a cell restricted to give rise to one group of hemopoietic cells. The committed PHSC are of two types. First, lymphoid stem cells which give rise to lymphocytes and natural killer cells. Second, colony-forming blastocytes, which give rise to myeloid cells. These are blood cells other than lymphocytes. When grown in cultures, these cells form colonies, hence the name colony-forming blastocytes. The different units of colony-forming cells are colony-forming unit erythrocytes. The stem cell of this unit develop into erythrocytes. Second, colony-forming unit granulocytes are monocytes. These cells give rise to granulocytes, which includes neutrophils, basophils and eosinophils are monocytes. Third, colony-forming unit megakaryocytes. Platelets are formed from these cells. Growth and reproduction of the different stem cells are controlled by multiple proteins called growth inducers. Interleukin-3 promotes growth and reproduction of virtually all different types of stem cells, whereas the others induce growth of only specific types of committed stem cells. Now we will see what actually this haemopoesis is. It's a process which involves the origin, the development and maturation of erythrocytes. The stem cells of this colony-forming unit E pass through different stages and finally become the matured RBCs. The various stages between stem cell and matured RBC are pro-erythroblast, which is also called megaloblast. Pro-erythroblast is the first cell derived from the stem cell CFU-E. It's very large in size with a diameter of about 20 micron and nucleus is large and occupies the cell almost completely. The pro-erythroblast does not contain hemoglobin. The cytoplasm is basophilic in nature. The pro-erythroblast multiplies several times and finally forms the cell of next stage called early normoblast. Early normoblast is the second stage. It's smaller with little diameter of about 15 micron. In the nucleus, the nucleoli disappear. Condensation of chromatin network occurs. The condensate network becomes dense, so this cell is also called basophilic erythroblast. This cell develops into intermediate normoblast. It's the third stage. This cell is smaller than the early normoblast with a diameter of 10 to 12 micron. The nucleus is still present, but the chromatin network shows further condensation. The hemoglobin starts appearing. The cytoplasm is already basophilic. Because of the presence of hemoglobin, it starts with both acidic as well as basic stains. So, this cell is called polychromophilic erythroblast. Fourth stage is late normoblast. The diameter of the cell decreases here further to about 8 to 10 micron. Nucleus becomes very small with very much condensed chromatin network and it's known as ink spot nucleus. In the late normoblast, the nucleus disintegrates and disappears. The process by which nucleus disappears is called pycnosis. The final remnant is excluded from the cell. Late normoblast develops into reticulocyte, which is the fifth stage. It's also known as immature RBC. It's slightly larger than matured RBC. The cytoplasm contains the reticular network or reticulum, which is formed by remnants of disintegrated organelles. Due to the reticular network, the cell is called reticulocyte. The reticulum of reticulocyte stains with supravital stain. The reticulocyte is basophilic due to the presence of remnants of Golgi apparatus, mitochondria and other organelles of the cytoplasm. During this stage, the cells enter the capillaries through the capillary membrane from source of production by diapedesis. Now the last stage, which is matured erythrocyte. The reticular network disappears here and the cell becomes the matured RBC. The matured RBC is biconcave and it's smaller in size with a diameter of 7.2 micron. It attains the biconcave shape and is with hemoglobin and without nucleus. It requires 7 days for the development and maturation of RBC from proerythroblast. It requires 5 days up to the stage reticulocyte, and reticulocyte takes 2 or more days to become the matured RBC. Now, what are the factors that are necessary for erythropoiesis? Development and maturation of erythrocytes require a variety of factors which are erythropoietin, thyroxine, haemopoietic growth factors and vitamins. Now we will see what actually leukopoiesis is. It's a development and maturation of leukocytes. The committed pluripotent stem cell give rise to leukocytes through various stages. 2 major lineages are formed, the myelocytic and the lymphocytic lineages. The granulocyte and the monocyte are formed only in the bone marrow. Lymphocytes and plasma cells are produced mainly in the various lymphogenous organs, including the lymph glands, spleen, thymus, tonsils and various pockets of lymphoid tissue elsewhere in the body, especially in the bone marrow and in so-called Peyer's patches underneath the epithelium in the gut wall. The white blood cells formed in the bone marrow, especially the granulocytes, are stored within the marrow until they are needed in the circulatory system. Now, what are the factors that are to be needed for leukopoiesis? Leukopoiesis is influenced by haemopoietic growth factors and colony-forming growth factors. Colony-forming growth factors, CSF, are proteins which cause the formation of colony-forming blastocytes. These are of 3 types. Granulocyte, CSF, secreted by monocytes and endothelial cells. Granulocyte monocytes, CSF, secreted by monocytes, endothelial cells and T lymphocytes. And monocytes, CSF, secreted by monocytes and endothelial cells. Now friends, let's learn about hemostasis. The term hemostasis means prevention of blood loss. Whenever a blood vessel is severed or ruptured, the injury initiates a series of reactions resulting in hemostasis. It occurs in 3 stages. First, vasoconstriction. Second, platelet plug formation. And third, coagulation of blood. First, vasoconstriction. Immediately after injury, the blood vessel constricts and decreases the loss of blood from damaged portion. The vasoconstriction is purely a local phenomenon. When the blood vessels are cut, the endothelium is damaged and the collagen is exposed. The platelets adhere to this collagen and get activated. The activated platelets secrete serotonin and other vasoconstrictor substances which cause constriction of blood vessels. The adherence of platelets to the collagen is accelerated by von Willebrand factor. This factor acts as a bridge between a specific glycoprotein present on the surface of platelet and collagen fibrils. The platelet aggression is also accelerated by another factor called platelet activating factor, which is secreted by neutrophils, monocytes and platelets. Second stage is formation of platelet plug. The platelets get adhered to the collagen of ruptured blood vessel and secrete ADP and thromboxane A2. These two substances attract more and more platelets and activate them. All these platelets aggregate together and form a loose temporary platelet plug or temporary hemostatic plug, which closes the vessel and prevents further blood loss. And third stage is coagulation of blood. During this process, the fibrinogen is converted into fibrin. The fibrin threads get attached to the loose platelet plug, which blocks the ruptured part of blood vessels and prevents further blood loss completely. Now friends, we will discuss the term coagulation and its mechanism. Coagulation or clotting is the process in which blood loses its fluidity and becomes a jelly-like mass few minutes after it is shed out or collected in a container. The process of clotting is initiated by platelets. Inside an intact blood vessel, the blood does not coagulate due to the presence of anticoagulants, heparin and antithrombin. There are different factors that are involved in blood clotting. Coagulation of blood occurs through a series of reactions due to the activation of a group of substances. The substances necessary for clotting are called clotting factors. There are 13 clotting factors that are involved in this process. Factor first is fibrinogen, second prothrombin, factor third is thromboplastin, factor 4 calcium, factor 5 labile factor, which also called proacceleratin or accelerator globin, factor 6 acceleratin, 7th stable factor, factor 8 antihemophilic factor, which is also called antihemophilic globin, factor 9 Christmas factor, factor 10 Stott factor, factor 11 plasma thromboplastin antecedent, factor 12 Hegman factor and factor 13, which is called fibrin stabilizing factor. Clotting of blood occurs in 3 stages. First stage, formation of prothrombin activator. Second, conversion of prothrombin into thrombin. And third, conversion of fibrinogen into fibrin. First stage, formation of prothrombin activator. Prothrombin activator is formed in 2 ways. Extrinsic pathway. In this, the formation of prothrombin activator is initiated by the tissue thromboplastin. And second is intrinsic pathway. In this, the formation of prothrombin activator is initiated by platelets which are within the blood itself. Extrinsic pathway for the formation of prothrombin activator. Tissue thromboplastin initiates this pathway. After injury, the damaged tissues release tissue thromboplastin. The thromboplastin contains proteins, phospholipids and glycoprotein, which act as proteolytic enzymes. The glycoprotein and phospholipid components of thromboplastin convert factor 10 into activated factor 10 in the presence of factor 7, which is stable factor. The activated factor 10 reacts with factor 5 and phospholipid component of tissue thromboplastin to form prothrombin activator. This reaction recurs the presence of calcium ions. Now, intrinsic pathway for the formation of prothrombin activator. During the injury, the blood vessel is ruptured, the endothelium is damaged and collagen beneath the endothelium is exposed. When factor 12 comes in contact with collagen, it is converted into activated factor 12 in the presence of kallikrein and high molecular kinogen. The activated factor 12 converts factor 11, that is plasma thromboplastin, into activated factor 11 in the presence of HMW kinogen. The activated factor 12 activates 9th factor in the presence of factor 4th, that is calcium. Activated factor 9th activates factor 10 in the presence of factor 8 and calcium. When the platelet comes in contact with collagen of damaged blood vessel, it releases phospholipids. Now the activated factor 10 reacts with platelet, phospholipid and factor 5 to form prothrombin activator. This needs the presence of calcium ions. Factor 5, that's labile factor, is also activated by positive feedback effect of thrombin. Now the last stage, which is conversion of fibrinogen into fibrin. Here thrombin converts fibrinogen into activated fibrinogen due to loss of two pairs of polypeptides from each fibrinogen molecule. The activated fibrinogen is called fibrin monomer. Fibrin monomer polymerize with each monomer molecules and form loosely arranged strands of fibrin. Then these loose strands are modified into dense and tight fibrin threads by fibrin stabilizing factor in the presence of calcium. Now, what is the role of vitamin K in blood clotting? Vitamin K is necessary for the synthesis of prothrombin in the liver. If vitamin K is inadequate in the diet or is not absorbed in the intestine, blood clotting becomes inefficient. This produces symptoms similar to those of hemophilia. Now we will learn about blood transfusion. Blood transfusion is the process of receiving blood products into one's circulation intravenously. Transfusions are used for various medical conditions to replace lost components of the blood. Blood transfusions typically use source of blood, one's own or someone else. Blood is most commonly donated as whole blood intravenously and collecting it with an anticoagulant. While blood transfusion, donor must be healthy without any diseases like the sexually transmitted disease such as syphilis, the disease due to virus like hepatitis, acute immune deficiency syndrome, that's AIDS, and only compatible blood must be transferred. Now, what is the procedure for blood transfusion? Before a blood transfusion, there are many steps that are to be taken to ensure quality of the blood products, compatibility and safety to the recipient, like the apparatus used for transfusion must be sterile. The temperature of blood to be transfused must be same as body temperature. The transfusion of blood must be slow. The sudden rapid infusion of blood into the body increases the load on the heart, resulting in many complications. Blood is transferred by exchange transfusion, which involves the removal of patient's blood and replacement with fresh donor blood or plasma. It's otherwise known as replacement transfusion and is an important life-saving procedure usually done in conditions like severe jaundice, sickle cell anemia, etc. Conditions which are to be needed for the transfusion of blood. Hemolytic disease of newborn, which is called as erythroblastosis fetalis, in severe sickle cell anemia, in severe polycythemia, toxicity of certain drugs in severe jaundice in newborn babies which does not respond to ultraviolet therapy. With this, we come to the conclusion of today's topic. Thank you for watching.